I welcome you all for today's webinar on where we are going to focus on accessing fine-grained molecular interaction data in IntAct database. I am Kalpana Paneerselvam, working as project lead as EMBL-EBI. Okay, the today's session outlined into three parts. Firstly, we are going to have an introduction to protein-protein interaction networks. And from there, we'll move on to IntAct, where we will explore multiple search options, and we are going to generate a network, and we are going to explore advanced filter options, look into interaction viewer for final details. And lastly, we are going to touch upon protein-protein interaction data integration challenges with context onto IMEx and PSICQUIC. Okay, protein-protein interactions are we call physical and selective contacts that happen between pairs of proteins in certain molecular regions and in a defined biological context. So interactome is the totality of all the interactions that happen in a cell or in an organism in a biological context. So protein-protein interaction network is the graphical representation of group of protein-protein interactions in which proteins are represented as nodes and the interactions or the relationship among the proteins are represented as edges. So why do we have to look at protein-protein interactions? So we all know that information is stored in DNA, it is transcribed into RNA and later on to protein. And proteins carry out all the functionalities in cellular context. So it's not that one protein does a single function. The proteins can perform multiple functions by interacting with multiple partners. Like given example in here, we have beta-arrestin. In the first network, we have beta-arrestin interacting with NK tumor recognition protein, which is a component of putative tumor recognition complex involved in the function of NK cells. In the network below, we have the same beta-arrestin 2 interacting with rho-guanine nucleotide exchange factor 12, which may play a role in regulation of Rho-A GTPase by guanine nucleotide binding alpha 12 and 13. So looking at this, we can understand that there will be two different functions because of different interacting partners involved in this context. So protein-protein interactions helps us to predict biological function of an unknown protein, so which we call as guilt by association, wherein proteins with similar functions are expected to cluster together. And they also help us to improve our understanding of protein complexes and pathways in a way, because the interaction networks work as a draft map or skeleton that brings detail to biological processes and pathways. So what we have here in the left side is 20S proteasome, which is a large multi-subunit complex containing 20S proteolytic core particle and pair of 19S regulatory particles. So this work it has been taken from Promega UK group, which used protein-protein interaction detection methods, particularly pull down combined with mass spectroscopy to detect all these components. And also they did protein-protein mapping experiments to find out the binary interactions. And towards our right, what we see is a beautiful work done by Glab et al., wherein they devised a methodology for extending predefined protein sets representing cellular pathways and processes by mapping them onto a protein-protein interaction network and extending them to include densely interconnected interaction partners. Thus, the added proteins display distinctive network topological features and molecular function annotations and can be proposed as putative new components and or as regulators of the a communication between different cellular processes. Okay, so there are multiple ways of experimental methods, multiple types of experimental methods that are being used to detect protein-protein interactions. Two methods are important due to their use in high-throughput studies. These are the yeast to hybrid complementation assay and tandem affinity purification followed by mass spectroscopy. Yeast to hybrid assay is a fast, inexpensive, and mostly it is a scalable method to detect binary interactions. Tandem affinity purification mass spectroscopy will detect, say, a multi-protein interactions, what we call as N-array interactions. There are other interaction detection methods that are less amenable to high-throughput setups, but they are used in small-scale studies. But they provide more detailed structural and chemical information about the interaction. So even though we have so many methods available to detect the interactions, we can say that no single method can accurately reproduce a true binary interaction, what is happening under natural physiological conditions. Basically because different experimental setups use different sample preparation types, and we may end up in bringing, for example, like we might be lysing cell, and so that there might be a disturbance in the cellular compartments and two proteins which are not really meeting in natural environmental condition, but may be brought to close proximity and thereby getting an interaction data out of it. So that's why we are depending on, say, not a single method, but we are depending on multiple pieces of evidences when you're trying to look into the, trying to look for more confidence on interaction data being generated. Okay, so protein-protein interaction, studies, they also give more details on the interaction domains. I mean, the definition of interacting interfaces. So we can specifically map them to the residue level. The domain can be represented as specific range of underlying amino acid sequence. So when a single protein is able to interact with multiple interaction partners, means it has different binding domains for each partner, and while representing this information in databases, we can try to map them to UniProt sequences. Sometimes there might be interesting post-translation modifications which are required for an interaction to happen. So this can also be mapped to underlying amino acid sequence. And while considering the representation of interaction data in databases, there is one problem with the complexes, because some experimental methods generate complex data. For example, the tandem affinity purification, so it brings multi-protein interaction which are associated. So we cannot directly bring in this information into database. We need to translate this association into binary evidences. So there are two algorithms available to transform this information into binary data. One is the matrix expansion model, and the other one is the spoke expansion model. In matrix expansion model, we have all the proteins are assumed to interact with all the available partners. And in spoke expansion model, one protein, usually the bait, is assumed to interact with the rest of the partners. Matrix expansion model generates a lot of false positives, and it becomes complicated. It generates a lot of data, and it is really complicated to maintain such level of, such dimensional information in a database. The spoke expansion model produces less noise, but in both cases, they may not be representing the accurate biological information, which is happening in the physiological condition, but we have to take a compromise here. Okay, so that is the introduction, small introduction that I wanted to give. From there, we would like to move on to IntAct. So IntAct is a publicly available molecular interactions repository. We have more than 1.19 million binary interactions captured from more than 23,000 publications. We capture only experimental data. So IntAct is hosted in EMBL-EBI. We are the founding member of the IMEx consortium. IMEx consortium is nothing but collaboration between public repositories, protein-protein interaction repositories, including databases such as MINT, MatrixDB, UniProt, IID, InnatedDB, DIP, etc., coming together and agreeing for a common curation strategy and following common curation standards for avoiding redundant data, for expanding more coverage, and also enabling data exchange between multiple partners. So we follow something called the deep curation model, capturing most of the experimental details in the interactions. And well, every curator, so there is always a double curator process to check for the maintain the quality. So such data, curated information is available for download via FTP, via website, and via PSICQUIC web services. So our sources are basically from 3 different sources. We capture the experimental evidence published in the scientific literature. And also we encourage direct submission from the authors because it includes, so it ensures the dialogue between the authors, authors and the curators. And so it helps us to represent factually correct and optimally aligned, representation of interaction data according to the author's view. And so this is something very interesting. And apart from that, we also get large-scale data sets from high-throughput projects. Okay, so the deep curation model allows us to capture details upon the host organism, the participant, and the interaction detection method, and also details on the binding kinetics, the stoichiometry data, and the external influences like the agonist and antagonist, which do not actually participate in the interaction, but they are present in the external, environmental setup, and they are known to influence the interaction outcome. So otherwise, there are the construct details, including the mutation information, which are mutations which are known to influence the interaction outcome, the known binding domains, and the affinity tags, link tags, and experimental preparations, which allow us to differentiate if the samples are considered as the physiological conditions or in any other artificial experimental setup, etc. Okay, so leveraging on the deep curation model, we have generated contextual interactomes. One good example is the mutations dataset, wherein mutations, which mostly the substitutions and deletions that are known to influence the interaction outcome, are being captured, mostly from low throughput publications. We have more than 75,000 annotations, and out of which we have more than 6,000 annotations that are associated to known rare diseases. And we have more than 50,000 annotations mapped onto Homo sapiens, and we have an automated pipeline performing sanity checks and to ensure that the sequence changes are updated in line with the latest UniProt KB release. And also, we have also and data sets contextualized on basis of diseases. So the latest one is the coronavirus data sets, wherein we have more than close to 14,000 binary interactions coming from more than 129 members of Coronaviridae family. So the data is constantly shared with EMBL-EBI's COVID-19 portal. We have mostly protein-protein interaction data, but sometimes we have, the nucleic acids and sometimes the small molecules also involved in these interactions. Okay, there are other data sets which are available in our download tab. So from here, I would like you to take to the live demo of IntAct database. Give me a minute. Okay, so we have landed on, this is IntAct homepage. So from the homepage, you have direct access to IntAct's COVID-19 data set, and also the information on the species-specific interactions linked to the species-specific interactomes. I mean, these are top 16 interactomes, species-specific interactomes are clustered, and we have direct links from our homepage, and you still have access to other data sets which are curated, computational and mutations data set and everything. And also linked to our FTP site and other, the training links that so far we have provided, maybe links to the webinars and other presentations that are linked to the training tile. And so I would like you to, just before performing basic searches and everything, so there are furthermore details about us in our About Us page, and there is syntax statistics, which allows us to keep up-to-date information on the number of interactions that we have captured on. So it was also segregated based on whether these are spoke expanded or binary to binary interactions. And of course, the number of publications and experiments that has been captured up to date, curation by reason, whether it is directly submitted or it is curation request by an author, or it is because of a funding priority or by curator choice. And the interesting part is that we have documented the totality of, interactions by the documented interaction detection method in IMEx, in IntAct. So as you can see, like the two hybrid array and the child terms of the two hybrid, so validated two hybrid and two hybrid rate pooling approach and two hybrid have the most share of, highest number of interaction. This is a interaction detection method documented in IntAct. And also we have the proteome coverage for the top 10 species. So these proteins are mapped to reviewed proteome of uniprot sets. So for example, in Homo sapiens, so we have, 86% of the reviewed proteome has at least one or more interactions documented in IMEx, and likewise for other organisms. And this is a summary of, all the features found in IntAct. So I thought I would take a quick tour before we start performing any searches in IntAct. Okay, so time to start with search, exploring the search options. As you can see, IntAct is supporting quick search, batch search, and advanced search. We are going to look one by one. So we'll start with quick search first. So in quick search, all the proteins in IntAct are backed by uniprot accession numbers. So they are our unique identifiers of any protein. So if we are not aware, if we do not know the uniprot identifier of the protein of your interest, and if we are generally in exploratory mode, so we can search in generic identifiers like gene names. So when you are searching for generic identifiers, there will be like a lot of options, a lot of interactors having the similar, getting the same, queried, fetched from the gene query. So in IntAct, we have this, there is this so-called auto suggestions that helps you to list out all the interactors for the generic query term that you have queried for. So as you can see, I've started queried for amyloid beta precursor protein, which is the gene name is APP here. So there are a lot of hits there are differences in the species name, but also there's something interesting that we would like to know. So there are, isoform specific interactions, and there are pro chain specific interactions. So we do capture, the species, the isoform specific and the pro chain specific information when we have relevant experimental evidences available. And so now for APP query, IntAct says there are 111 interactors, different interactors, now listed on first page, and we have in second page, and etc. So for our query, let me pick up something nice. Okay, so let us say I'm interested in prochain 40, APP prochain 40. Okay, so now when I select that, I can also choose to hit the search option. It will bring all the interactions. So here I just chose one of the proteins as my interest to actually explore the network viewer and also the advanced filter options here. Okay, so now we have landed upon the results page, first supported by the network. So now let us have a look at the legends. So the legends indicate so there are the nodes are, you have different color. The nodes are color coded according to the species. So we have at least like Homo sapiens and Mus musculus as major species, and there are other animals and other mammals. And as you can notice, the edges, which represent the relationships, so they are color coded, and also there is a difference in the width. So what does that indicate? So actually, this view is in summary view. If you are looking at any edge, you can see there are, for example, in this edge that has been hovered upon, so it shows there are three interaction detection method evidences behind this edge. So means that, so this is the default summary view. And what is a summary view? Basically, behind every interacting pair, all the evidences that contribute to this interacting edge are summarized into single edge. So here in this case, so the highlighted, the edge that has been pointed now using the cursor. So now it says it has three interaction detection methods behind three pieces of evidences behind this edge. And likewise, we have another edge with higher thickness, higher width. So that has 4 interaction evidences. And you can also notice about an MI score. So that is, and here the legend says that your color intensity is backed by MI score. So what is MI score? So MI score is a short for molecular interaction score. So when I was talking about the in my previous slide, I was mentioning that no single experimental evidence can actually replicate the true biological binary interaction happening in a system. So because they can be artificial, so artifactual. So to increase the confidence behind every interacting pair, IMEx has come up with an MI score, which depends on 3 major factors. The first one is the type of interaction, means like whether they are found to be physically interacting, or they are, found to be in close proximity or just co-localized, something like that. Then based on the number of evidences and also based on the type of interaction detection method. So combining these three factors, we have an algorithm that can generate an MI score. And so if you ask me what will be the threshold that you have to set for looking for the high confidence interactors, I would say the threshold should be set up at say 0.49, which means that you have at least 2 evidences backing up for a particular interaction. Of course, so when there is a filter for MI score, and so right now the filter is activated. I have set a threshold for 0.49, and you can see only the high confidence interactors are listed in our network. So you can choose to remove the filter. Okay, so this is about the summary view. And there are other options to, you can also choose to expand used to see the expand view, wherein all the edges, so every evidence is represented as a single edge, and you can see there are a lot of evidences for APP self-interaction, and there are several edges between every interacting pair. So if you hover around the node, it will give you the node level information about the protein and about the species of it, species of origin of the node. And if you hover around the edge now, so it gives you since every evidence is expanded as a single edge, now we know about it shows the type of interaction detection method behind every edge, right? So yeah, so this is about, this is in the expanded view. So there is another view, which is interesting. Again, so affected by mutation. So in this view, we are still at the expanded view, but the node and the affected edges, the edges that are affected by the mutation are highlighted in pink. So yeah. And so now let us, okay. So you can choose to export the image in PNG or GraphXML format, and also there is an option to export the tables. So we haven't looked at the table. Let us see what the information is available at the tables. So all the information, behind every edge is listed in the tables. Now you have 2 tabs. You can look at the information at the interactions level or also at the interactor level. So, you can see the tab, tables are paginated. And of course, you have at least 40 columns, and there's a default columns that are selected for the view. And sometimes if you are interested, or if you feel that these columnar information are too complex and you don't want many of this information, you can uncheck them and to see what information, or you can select all this information for you to view the details or whichever column you want to. So this also applies for interactors as well. And you can download the table in MyTab or XML or MIJSON format. Okay, so now, okay, let's have a look on the layout. So the default view is the force layout. Now let me uncheck the mutation view and the expanded view. Okay, so there's also a circular layout that people might be interested to look upon. Also, we can also choose the bubbles layout, wherein the common species are put in one bubble, and they are segregated according to the species. Okay, so now let's get back to the force directed view. So yes, so and also you can choose to, within the network, you can choose to query for a protein if you are interested in querying for a protein. So you can query here, and if it is available, yeah, it will. So you can hit the search option, and it will highlight the protein of your interest. Okay, so yeah, so this is about when you are in the exploratory mode, and when we are using G name as the identifier for our query. Okay, so I'd like to do another query. So with using UniProt identifier. So when we are searching with unique identifiers, so for example, I have chosen an identifier which stands for the mitochondrial antiviral signaling protein. So now again, it is very specific, but still it brings two hits. Again, one for the canonical form of the protein and another one is the isoform 1 version. So here I'm not specific about any specific version. I would like to go for the hit. So I'm hitting the search option. Now IntAct servers are queried for this identifier and I get a network, and I can still play around with this network to move the nodes along and etc. So, and what I would like to do now is to, so now let us explore the filter options. Yeah, so here, the first filter is the interactive species. So now we have, so what is displayed in the menu is like all the species, the available species of the interactors. So the filters allow you to choose a specific interactor. For example, if you're looking for interactors, at least one interacting partner belonging to Homo sapiens, you can choose to select this filter. And then, give me a minute. Yeah, so now one interacting partner is belonging to Homo sapiens. So if you're looking for only interactors between Homo sapiens and Homo sapiens, then you can check this option. So that will restrict you to ask the network to display only those interactors belonging to one species, that is Homo sapiens. So you can look at the nodes that it has changed, filter has been changed. Okay, so you can remove the filter option, and this can apply to any other species. And is another filter that interactor type. So in this network, we just have all the interactors are proteins. So sometimes if there are occasions wherein mostly we have protein-protein interactions, but sometimes there are interactions coming having nucleic acid or small molecule as an interacting partner. So in that case, you can filter out if you're not happy with having long coding RNAs or any other nucleic acid type. If you do not want, you can activate such filter for choice. Then the important filter is I would say is interaction type filter. Sometimes so there are multiple types of interactions. Interactions of physical association, like East2 hybrid, generate these types of binary interactions. And association is like maybe the tandem affinity purification mass spectroscopy is a good example, wherein an interaction detection method that produces association data. And co-localization is, wherein the proteins are not interacting actually, but they are found to co-localize, say by, as detected by confocal microscopy. And proximity is where the proteins are found to be proximally close, backed by bio-ID assays. And direct interaction when the proteins are purified and the interaction is detected in vitro or specific ubiquitination assay. And you have filters for different types of interaction detection methods, and the same applies for host organism. Sometimes if you are looking for interactions that are happening not in lab-grown cell lines, you can choose to filter out those. In our case, we do not have much options that way. And this is another filter where you can choose to look for interactions that are affected by mutation only. And this expansion filter. So this expansion filter filters out all the, it's an option that is available. You can choose to filter out all the spoke expansion interactions, thereby looking at true binary interactions within your network. And this is a filter, positive filter. So it says positive and or negative interactions only. So I mean, negative interaction. So by looking at the name, the negative interactions, we can safely assume that there is no interaction. I mean, negative interactions mean technically there is no interaction between them. You might be wondering, like, why will IntAct curate such an interaction and say there's a negative interaction? Well, there is a specific context. So many times, I mean, not many times, but in rare cases, there might be cases wherein one specific isoform of a protein is known to interact with another partner. And there might be other isoforms which are of the same protein not known to interact with the other partner. So in those cases, backed with proper experimental evidences, we capture interactions. I mean, this isoform specific interactions as negative interactions. And so these are very rare evidences. You do not have much interactions, but there are cases. So if you're not happy with inclusion of this negative interactions, you can choose to filter out. In our case, we do not have any negative interactions, so we are fine. And IMEx co-filter, as I had mentioned before, so this is to this filter allows you to select high confidence interactions. So I would like to activate my filter. So I would like to set my threshold to 0.5. So yeah, it takes a while because just a second. I can refresh. Yes. Yes, so now, yeah, sometimes it happens that you have to just refresh the page. Yeah, so now I have only high confidence interactions. And yeah, so now again, I would like to look for those associated with mutations. And yes, so I can choose to look at any, so now I have information for all the, the table displays interaction evidences behind all these, interacting partners within this network. Okay, so yes. So in my case, let me see. Okay. I think I would like to maybe, yeah. Maybe you should have more interactions. Yes. Yeah. Okay. So now, yes. So now again, so there are few interactions with mutation information, and there are interactions which are spoke expanded, and there are other interactions as well. So I can choose to look for any. I can select any edge and let us say if I'm looking for interaction between MAVS protein and TRAF6, so I have selected an edge which says that interaction detection method is, so the edge is detected by, it represents evidence detected by biolayer interferometry. And as you can see, the network table brings out information only for the edge that has been selected. And if I choose to click on this icon, so it takes me to a new page, which is actually the interaction viewer page. And while the default view of this viewer page is like you have the interacting pairs, and they appear in the collapse mode. I can choose to expand this protein and maybe up to the amino acid level. Sometimes it's interesting to see those features mapped to the amino acid sequence. And also, yes, yeah. And also you can choose to collapse them or expand them as much as you want. And yes, so this interaction viewer is a collaborative work of EBI with RAPS laboratory. And so this allows us to view the finer details of interaction. So basically, the default MI feature, molecular interaction features curated by IMEx curators are loaded by default in this viewer. And also you can choose to load with features captured in UniProt, and also fetch information from superfamily or any interactor. Okay, so, and coming down, again, you can choose to, export option is available at the interaction viewer. And coming down, you have the details. tagged, segregated as interaction details, participant details, participant tab, and features tab. So now, so it says we have a direct interaction, and the interaction detection method is biolayer interferometry. And if you're not familiar with what is this type of interaction, you can choose to click on this, wherein it will take you to our ontology, which gives you a definition of what is a biolayer interferometry as an interaction detection method. Same can be applicable for, if you're not clear about direct interaction, again, it will choose you, yes. As in, we have our ontology backed with, every control vocabulary that we have been using. Okay, so now we have the details of the interaction selected, at the interaction level. So it seems to have a binding, KD data available for this interaction, which is 36 nanomoles. Okay, so, and you have information available at the publication level. And also when you like to move on to the participants tab, so now we have information on the interacting partners and what is their role. So TRAF6 takes the bait role, and MAVS protein assumes the role of the prey, and there is no identified biological role in this case. Okay, and they are expressed in E. coli. Okay, so then you can choose to look at the features tab, which is telling you that. Okay, so now we have two features for TRAF6 protein. One is the sufficient region, which is ranging from amino acid positions 310 to 504, and it also carries a biotin tag. And MAVS protein is found to have 4 interactions, 4 mutations, 2 at amino acid 460 and 1 in 457 and 455. So yeah. Okay, I think it has been superseded by type of information that we have looked upon. Yes. So yes, so we have interaction as 455 and 457 and 460. Yes. So these interactions, so we have one mutation, one substitution mutation at 460, tyrosine 460 substituted by alanine is known to disrupt the mutations, and you have another disrupting mutation at 457, and of course another disrupting mutation happening at 455 as well. So apart from this, you have MAVS protein is known to have another sufficient to bind region ranging from 449 to 467, and you have this protein has a GST tag as at the N terminus. Okay, so we have explored the interaction viewer, and let's get back to our IntAct search. So that's it about the quick search. So by moving to the batch search. So yes, this batch search is for looking for large list of, big list of interactors if you'd like to query for, and it can have a heterogeneous mix of, identifiers. So for the sake of simplicity, for this example, I would like you, like to take 3 different identifiers, which I have chosen for this. So I'm using one UniProt identifier and a GTP, which could overlap between a gene or a small molecule and another gene name for this query. So you can also choose to upload the list of identifiers using a file using this upload file option, or you can choose to copy paste in this query box. So after pasting your list of identifiers, and I choose to go to the next step. So now it takes me to the disambiguation panel, because GTP has not only GTP has it has mapped a lot of interactors, so I have a lot to choose from. And similarly for LRRK2, and as you can notice, the UniProt identifier has fetched directly the direct unique identifier from it. So for GTP, I would like to unselect all because it's a huge bunch, and maybe I'll choose only for the GDP, which is guanosine triphosphate. And for LRRK2, again, I'm not happy with all the identifiers. Maybe I can choose to have these LRRK2 from rat, and I'm fine with this identifier. Okay, so once I've selected the unique, the interactor of my interest, I'm proceeding to search further. Again, so now I'm being taken to the network viewer, wherein you can see the results. Of course, GTP has a lot of interactors coming from a lot of species, so it's really colorful. The network is colorful. Okay, I think I need to refresh again to look for the interaction data. Yes, the tables. Yes, it takes time to load. Okay, so that's it about batch search. So moving on to the advanced search. So as you can see, advanced search is something like you can use our MIQL query syntax. There's a supporting documentation available. Sorry, so it takes me directly to the documentation here. So you can, it gives you the fields and what type of functionality, what type of query it can generate, and so on. So based on this, you can generate your query. So now I'll try to perform an sample query here. So as I mentioned, IntAct has a lot of data sets, and one data set is called as the rare disease data set. I would like to query within the rare disease data set, so that this all the data set are having an annotation type, and so I'm querying for a rare disease data set. So this is the rare disease data set name. And I would like to filter it for only the high confidence interaction. So I'm setting the IntAct score, IntAct score to say, 0.5 to 1.0. Okay. So, and we'll also like to see if, to include those ones having a feature type, having binding information on the binding regions, and or it can also have a mutation. I'm sorry. So this should be, yes. So okay, so this, as you can see, like whenever I try to start filling in the in this query box, so you can see the MIQL query builder actually replicates the query here, and it splits the query in as a subrule, subset of the query, and yeah. So now if I query, of course, I'm going to get a bunch of interactions. Oh, sorry, there is no interactions found. Something wrong with the query. Maybe I can try something else. Maybe I can look for those query for all intact proteins. Just query them for MI score. So that can be also done in zero. Okay, so maybe we can add something here. Let me select something. Okay, so let me say. So okay, let me look. So okay, so here that was the documentation. Let me have a look at the documentation. Okay, so there could be a biological role in interactor A or interactor B. Okay, so let me do something from here. So it could be a biological role. Let me see if there is any inhibitor, or is there any parameters in the interactions? So let me see. Okay, so now we have queried for IntAct for to fetch interactions wherein the one of the biological role of one of the interacting partners to be an inhibitor, and it might also contain, it can otherwise fetch all these interactions having a parameter. So yes, as you can see, there are more than 1,500 interactions in our results. So the network viewer is disabled because basically beyond 1,500 interactions. So you will not be able to process any information. Basically, the network viewer will be looking as like a huge, hairball. And so this is, as you can, now the export option is disabled for higher interactions. This is a work in process. Maybe in a couple of months or in some time, we would like to have this enabled for large set of interactions data. So this is what I wanted to show you for as an advanced search option. Yes, so now we have looked at almost accessing all the data, hidden information, not hidden, but the accessing all the multi-level information in IntAct. Okay, so now I think, yeah, now let's go back to our presentation. Okay, so the last part. So the last part is PPI data integration. So there are a lot of databases and interaction databases, and earlier there were many of them following different data format and standards, which is making the user very difficult to actually query. The user has to query multiple interfaces, format different query patterns, and the data that the data the person gets from each database could be carrying redundant information, and it will be very difficult to overlap or integrate any information that is fetched from each database. So this is the challenge when you have multiple databases operating in different data formats and standards. And there's always a challenge with standards, like when you're trying to unify a standard, we might accidentally create a new standard unwantingly. Okay, so there's a good compromise if you can allow different different databases to operate instead of a single database, but option to query through all the databases through a single query interface, and the data that you get from those interfaces can, will be if this is in the same standard, curation standard and format, it will be easy to integrate for the users and also to analyze information and so on. IMEx consortium was created in order to have a community standards between multiple databases. So IntAct is the founding partner of the IMEx consortium, wherein we agreed to follow the databases like IntAct, DIP, Mint, and MatrixDB, and so on. So in InEdDB, all agreed to follow the same data standards and same curation strategy. So you will, so our partners, so when you're querying any query through IMEx consortium or through IntAct, so you'll be able to get information from all these databases, and so they will not be redundant, and we have our own domain-specific coverage for the interactome, and eventually data is available for in MITab or XML formats for further analysis. You can integrate your data exchange and do your analysis you can query in IMEx or in IntAct. I'll show you in a bit using a PSICQUIC web service, which allows you to access all this database information fetched in all these databases. So this work is backed up by generating common identifiers, vocabularies, formats, and schema by PSIQUIC standards generated by Proteomics Standards Initiative. And yes, so this allows us to distribute data across our databases. So PSICQUIC query. PSICQUIC is our unified Proteomics Standard Initiative common query interface, which allows data to be disseminated. You can also query for using PSICQUIC. So this is the link is available here. So the same information you can PSICQUIC is another web interface wherein you can access similar information for our IMEx partners. So there is this is our PSICQUIC view tool. So these are the listed services, databases which choose to agree to data, allowing data exchange via PSICQUIC. And there is also a PSICQUIC registry. I'm sorry, this is the query results when you are querying for a any protein, say PF3, and here is like you can also select any database from this from the listed databases. So as you can see, there are databases listed in green are the ones which are active ones, and there are the gray databases which are not active at the time of our query. So there's a PSICQUIC registry that allows you to look at the status of the available databases which are active or not. Yes. So I think I've taken more time for completing this webinar. I'm sorry. So I think let's start having, if there are any questions, I would like to answer now. So with this, we complete our webinar.
Right. Thank you. Thanks a lot, Kalpana, for the webinar today. And thanks everyone for joining and also asking a few questions. All right, so with that, there are two very good questions, Kalpana, for you. So first one is how to make use of IntAct for non-model organisms. So this question came when you were presenting the interaction network after the search.
So IntAct is having, so we have more coverage on, of course, human proteome is our primary coverage, but we have, as I mentioned in my the previous slide, we have a lot of interaction. We have top 16 interactomes coming from, human, Arabidopsis thaliana, yeast interactions, E. coli, and several viruses. Mostly recently, we have a lot of information coming from coronavirus data set. And we have a lot of species, but you have at least like the top 16 species give you more, you have a lot of information recorded for the top 16 species, and that's why we have segregated at species-specific information to give you direct access. So if you are talking, there are information, minor information coming from different viruses, but we'd like to focus them elaborately right now with the resource constraint and also on the demands from the grant requests. We have different approach based on what is going to be the priority right now. But if you think that we are missing specific piece of information which should be actually, if you think that that should be part of IntAct and you are missing that publication, kindly mail me. You can directly, I have given my mail ID, you can contact me to that, or you can send us through the feedback and we'll be happy to take that in our priority list and have it processed for the immediate release.
Okay, thanks, Kalpana. And I think the person who asked the question has clarified that non-model organism is where I think they don't have the genomic information available.
So if the genomic, for us, we need the, we need uniprot identifiers. So for that, we need the genome information to be translated in protein identifiers. And without that, we cannot have any interactions in IntAct.
And another interesting question is, if somebody is using IntAct in their work or publication, how can they cite specific pages of IntAct?
Oh, yes. So I should take you to the, so for citing IntAct, and thank you for asking these questions. So if you are citing us, I mean, so you can go to our IntAct page, and this is where, so we have a recent publication for citation. I can share this link on the chat box. Yes, so you can use this publication for citing IntAct. Thank you for the citations, because like this gives us more visibility and also improves our chances for getting proper funding while we are applying for grant applications. Yes, thank you.